In this module, we will talk about Henry law and the oxygen supply. What is Henry law? Henry law actually describes the solubility of oxygen in nutrient solution. Another, we can say that the fermentation medium in relation to the oxygen partial pressure in the gas phase. So this equation, you can see that C star is equal to P naught over H. C star is the oxygen saturation concentration of the nutrient solution. So it is the amount of oxygen present in the nutrient medium. Then is the P naught is the partial pressure of the gas in the gas phase. When the oxygen or any that the percentage presence of the oxygen in the air or in the gas that we have to supply in that fermentation. So H is the Henry constant, which is specific for the gas and the liquid phase. So here the point to be noted that aeration with air, if we supply common air that we know that mostly contain 21%, if we say that the normal air having no pollution etc., then we can see that there is 21% of the oxygen in that. So if we aerate the fermentation medium, then that contain that capacity of 9 milligram of oxygen per liter dissolved in water. But when we say that pure oxygen, then that has the 43 milligram of oxygen per liter. So when we supply the pure oxygen, then there will be a more solubility, and then there will be a more provision of the oxygen into the fermentation medium. So this is the Henry law. So as concerned the oxygen supply, so the oxygen is normally supplied to the microbial culture in the form of common air, because that is the very cheapest way. So this method for the provision of a culture with the supply of air varies with the scale of process. So as we know, when the fermentation process, just we have a provision of a conical flask, which we called as shake flask. We have to meet the requirement of the oxygen just by shaking, and we can use maximum up to 250 to 500 ml just by the shaking on some platform with the controlled temperature. And we know that when we are dealing with the shake flask fermentation, especially in the conical flask, we can maximum utilize in case of aerobic fermentation, when we are dealing with the unicellulars, mean bacteria and blue-green algae, then we can utilize 1/5 of the total volume of the flask. But in case of the filamentous fungi, we can maximum utilize 1/10 of that. So if the flask is 250 ml, then in fungi we can use the 25 ml of the fermentation of the flask, and if there is the bacterial culture or the yeast culture, we can utilize 50 ml. So the remaining volume is used as a headspace that required for the transportation and then the exchange of the gases. But as concerned the pilot scale and the industrial scale fermentation, normally it is carried out by the stirred fermentation, or in a highly aerobic fermentations we can see that there is airlift fermenters or the tower fermenters. So the aeration and agitation is a problem, and we have to when we have a deep fermentations. So Bartholomew et al. Reported in 1950, they represented that the transfer of the oxygen from the air to the cell where that is consumed pass through 3 different steps. According to Bartholomew et al. 1950, there are the 3 different paths of the oxygen transfer. The first one, that the transfer of oxygen from an air bubble into the solution, and the second is the transfer of the dissolved oxygen through the fermentation medium to the microbial cell, and then the uptake of the dissolved oxygen by the cell. So these findings have shown to the correct when there is a non-viscous fermentation. but it has been demonstrated that the transfer may be limited by other stages in highly viscous fermentation. In later slide, we will see that what will be the possible path of the oxygen when starting from the air to the cells. If the cell is eukaryotic, mean that oxygen is needed in the mitochondria. You can see in this slide, then oxygen has to pass 9 different phases. You can see here that 1, 2, 3, 4, 5, 6, 7, 8 and 9. These are 9 different steps and the phases involved in oxygen from the air bubble to the mitochondrial site. So first is the air bubble, then is the gas liquid interface, then is the bubble layer, and then is a bulk medium, slime boundary layer of the cells, then there is cell wall, and then is the cellular floc or the slime, then is a mitochondrial wall. So 9 different steps has to pass from the air bubble to the cells or to the destiny where the oxygen has to utilized. In this slide you can see that how that the concentration in the gaseous form to go into Cx and then Cy into Cf and then Cf. Actually this is just the elaboration of the previous slide, then how the oxygen complete its path to its complete utilization. So the requirement of high dissolved oxygen concentration by many fermentation has resulted in the development of the process techniques to ensure to complete this transfer path does not exceed the oxygen supply capability of the fermentation vessel. So if the oxygen has to pass so number of phases, then how much each and every step is very critical. So in later module we will talk about the role of each and every step in the path of the oxygen from the air bubble to the cell where that has to utilized.